everybody. So moving on to chapter 20 this week, the lymphatic system and lymphoid organs and tissues. Super short chapter. We're just going to kind of get through this in one nice swooping lecture or video. Again, short week, or short, not necessarily short week, but short chapter this week, just this information. So the plan for this lecture is to literally go over the entire lymphatic system and lymphoid organs. And what that means, we'll talk about the lymphatic system and kind of the breakdown of what it is and how it functions. We'll talk about lymphoid cells and tissues. We'll talk about lymph nodes, and then we'll end with other lymphoid organs. So the objectives list is here again. Again, probably the shortest objectives list you guys have had in AMP1 and AMP2. But a nice short one for you guys. So make sure again you guys are looking over the objectives for the quiz that is going to correspond with this chapter. So when we talk about the lymphatic system, so last or 2 weeks ago we got done talking about blood vessels, and we talked about that notion of bulk flow. So we have, right, those arterioles that come in, and we have our capillaries, right, and we talked about the fact that we had this bulk flow out movement on one end, on the arterial end, and the bulk flow inward movement on the venous end. And we talked about the pressure that exists in the capillary and the pressure that exists in the interstitial fluid. And essentially that hydrostatic pressure of our interstitial fluid, if you remember, it was about 0 millimeters of mercury. Okay, and it was 0 millimeters of mercury because of the lymphatic system. Okay, so what the lymphatic system does is it returns that interstitial fluid as opposed to it being driven back into the vascular system, into the venous side of our capillary. Instead, as it's leaked out through that hydrostatic pressure, okay, from the blood vessels, it's actually kind of swooped up by the lymphatic system and brought back to the heart. Or not necessarily to the heart, but to the blood vessels before they enter the heart. So the lymphatic system consists of 3 major parts. It consists of our lymphatic vessels, which is very similar, which we'll talk about here over the next few slides. Very similar to our venous circulation. It is comprised of lymph, which is the fluid in the vessels, which again is very similar to plasma, but however slightly different. It doesn't have the plasma proteins in it. And again, we'll talk about that here in a few minutes. And then our lymph nodes. Our lymph nodes are basically the cleaners of the lymph. So this is basically the structure that lymph, as it goes up and being returned to the venous circulation, gets cleaned through those lymph nodes. Now, there's other lymphoid organs and tissues which will provide basic structural support for our immune system. And the reason it's able to provide that basic structural support is because it contains a lot of phagocytic cells as well as lymphocytes. So when we look at the lymphatic vessels and look at the distribution and the structure, unlike the cardiovascular system, where we have the artery to the capillary to the venous system, and it's a circular system, the lymphatic system is a one-way system. Okay, so what we mean by this is that there's not a circuit, there's not a circular system to it. Instead, the fluid's going to leave the capillaries, right, and it basically is going to enter into the lymphatic system, and again, be brought back to the heart. And so we can see that kind of here. We can see that arterial system here, right? This is what we talked about in chapter 18 and 19. We have more specifically the vessels in chapter 19. Here's our capillary system, and here that capillary basically is going to return the venous blood to the heart. Okay, and this green structure here now is our lymphatic system. So what's going to happen is that interstitial fluid is going to basically enter the lymphatic system via the lymphatic capillaries. Those lymphatic capillaries will then merge into collecting lymphatic vessels, and those collecting lymphatic vessels will then empty into lymphatic trunks and ducts. Okay, so let's first look at the lymphatic capillaries. These are what we call blind-ended vessels, which means they just kind of have like a dead end to them. They're not continuous like we think about when we think about vessels. and capillary systems. And to hold these lymphatic vessels kind of in place, what they have are these little filaments that anchor them to the connective tissue around them. And in addition to that, they have these really, really significant microvalves. Okay. And what these microvalves allow for is massive movement of fluids. So these types of lymphatic capillaries are actually super permeable, which means it can take up proteins from that interstitial space. It can take up cell debris from that interstitial space. But what it also means is that if you have, you have your cell, right? And if you have bacteria or a pathogen in that interstitial space, it is also going to take up the pathogen or even cancer cells. So when they talk about cancer cells metastasizing or moving from a primary location to a secondary location, typically what they'll do is they have the cancer's primary site, and then they check the lymph nodes surrounding that primary cancer. And that is because typically the way cancers are able to migrate to different parts of the body is through the lymphatic system. Okay. So again, lymph is going to enter into these lymphatic capillaries. Okay. And then they'll enter into the larger lymphatic vessels. So those lymph capillaries will drain into the collecting lymphatic vessels. Okay. These are very similar to veins, except that they're a much thinner wall, and they have more of those internal valves to help that lymph migrate up or move up to the heart. And it does have a decent vasorum that's going to help supply the nutrients that it needs in order to carry out its function. So we have the lymphatic vessels, collecting vessels, which then will basically come together and form these larger trunks. And the trunks are actually paired. You have a lumbar trunk, so you have a right and a left lumbar trunk. You have a bronchiomediastinal trunk. You have a right and left. You have a right and left subclavian trunk, a right and left jugular trunk, a right and left interstitial trunk. Okay. So again, remember, we're moving up into these different systems, right? And we can kind of see that here. Here's my, right? We can kind of see that here. Here's our capillary way down here, okay? And these move up into our collecting vessels, right? And all these vessels then empty into our collecting ducts, our trunks, okay? So these are trunks here, which will ultimately then dump into our ducts. So we have two large ducts that all of those trunks will empty into. Okay, so we have our lymphatic capillaries, to our lymphatic collecting vessels, to our lymphatic trunks, and all the trunks will then dump into one of two lymphatic ducts. They'll either dump into the right lymphatic duct, and this basically is going to have lymph from the kind of right upper arm and right side of the head and thorax will dump into the right lymphatic duct. The rest of the body will dump into the thoracic duct. Okay. Now, each one of those will empty its lymph into venous circulation at the junction of the internal jugular and the subclavian veins on both sides of the body. And we can see that here. Okay. So what we can see, for example, is we can see this lymphatic network, okay, and we can see kind of our collecting vessels here are going to basically empty into our different trunks, okay. Here is our thoracic duct that comes up here, okay, and this is going to come up and it is going to empty into one of the two duct, or into the right or left into the internal jugular or subclavian veins on their own side of the body. Okay. So again, this is kind of the network. And what we can see is, for example, this distribution of kind of lymph, right? We can kind of see this split down in this light green area. Okay. So this whole region here will dump into the right lymphatic duct. All of the red and the rest of the body will dump into the thoracic duct, okay? And again, this will come up, and for example, the thoracic duct will empty in to the subclavian over here, okay? And the right lymphatic duct will enter into the jugular vein on the right side. So how lymph is transported, again, it's a low pressure environment, just like our venous side, okay? However, and the way it's propelled back to the heart is very similar to the way in which our venous blood is returned to the heart as well. So lymph is propelled by a number of mechanisms. So one, the milking action of the skeletal muscle, which is again just like the venous system. Pressure changes in the thoracic cavity, just like the venous system. Okay, we have valves to prevent that black flow, just like the venous system. The lymphatic system can kind of hijack the pulsation of the arteries to help kind of propel it through the lymphatic system, as well as contraction of the smooth muscles in the walls of the lymphatics as well. So again, very similar mechanisms of transport that we see in the lymphatic system that we saw in the venous system as well. So when we start looking at the cells that are kind of make up the lymphatic system, it consists of kind of two populations of cells. One are our immune system cells that are found in the lymphoid tissue itself, and the other are the supporting cells that are going to form the lymphoid tissue structures themselves. So we're going to move into next week talking about the immune system. So chapter 20 and 21, if I were to do a face-to-face lecture, are typically combined in the same week. Because you guys are just coming off of exams and have an exam this week, I separated them out to have a lighter lecture this week going into next week's or this week's lab exam. So we have our lymphocytes, which we talked about in chapter 17, which are our main warriors of the immune system. Again, they're all going to rise or come from our red bone marrow through that process of hematopoiesis. And when we look at our lymphoid cells or our lymphocytes, we get actually two cell populations out of that lymphocyte population. We get what's known as our T cells, also known as T lymphocytes. So typically when we talk about lymphocytes, again, we have our B lymphocytes and our T lymphocytes. However, in terms of immunology, they don't ever really refer to them as T lymphocytes and B lymphocytes. They typically refer to them as T cells as well as B cells. Okay, so that's typically how I refer to them, is kind of talking about them in the aspect of T cells and B cells. So again, but remember, both of these are a type of lymphocyte. So T cells are really there to manage our immune system. They will attack and destroy infected cells. Our B cells are there. They're going to produce or differentiate into, once they become activated, into a cell known as a plasma cell. And the function of that plasma cell is to secrete antibodies. Again, this notion of antibodies should be somewhat familiar to you guys. We talk about antibodies in our ABO blood typing. So we'll talk a lot more about antibodies next week when we get into chapter 21. But essentially what antibodies are going to do, they're going to mark those antigens for destruction. And once that pathogen, which is what's going to be on an antigen, which is going to be on a pathogen, so that's what we're marking. Once that antibody binds to that pathogen or the antigen on the pathogen, that is going to trigger a phagocytic process and cause that pathogen essentially to be destroyed. So other lymphoid cells that we'll find within the lymphatic organs, we'll have our macrophages. Okay, so again, remember, our macrophages are essentially monocytes that have left circulation and entered into the tissue area. These will also help activate our T cells. Again, we'll talk about next week in chapter 21. Our dendritic cells, these are going to capture our antigens and deliver them to the lymph nodes. They're kind of like you can think of it, right? You have a bad guy, right? And you have a mugshot of the bad guy, and the dendritic cells are like the ones that go to where all the cops are and show the cops like the photo, and say this is who we're after. Okay, so that's kind of like the dendritic cells. They're what we call antigen presenting cells, and again, we'll talk about that next week in chapter 21 a little bit more. Then we have our reticular cells. So these two here, our macrophages and our dendritic cells, are really involved in an active fight or part of the immune system response. Okay. However, our reticular cells, which are really, really important as well, they're not necessarily involved in the immune system response. Okay. They're more involved in producing or providing the scaffolding within the lymphatic system where all these cells are able to reside. Okay, so again, because remember, we have our T cells, we have our B cells, we have our macrophages, we have our dendritic cells, and I know I keep on alluding to chapter 21 a lot, but all of those are going to actually carry out that immune function in our lymphoid tissue. Okay, and without the reticular cells, we can't generate that reticular connective tissue that our lymphoid organs need in order to support the cells of the lymphatic/immune system. So the lymphatic tissue and organs themselves provide a structural basis of our immune system, okay, which is essentially what I just said. They house our phagocytic cells and our lymphocytes, and their structures include things such as the spleen, the thymus, our tonsils, as well as other tissues known as lymphoid tissues, and these tissues can be fairly diffuse. So when we talk about lymphoid tissue itself, so like a spleen makes sense, right? It's an organ. We have, right, our thymus, which is an organ, right? Or a gland. But we also have these tissues that, and when we talk about tissues, typically they can be either localized or they can be diffuse. And again, I'll talk about what I mean by that here in a minute. So the tissue, our lymphoid tissue, houses and provides an area where our lymphocytes, remember these are our T's and our B cells, can divide or proliferate. So proliferate, this just equals divide. Okay, so remember these are our T's and our B cells here. Okay, and so what this lymphoid tissue does is it really provides an area of surveillance, okay, for these lymphocytes and these macrophages. Because again, like, you have all of your cops patrolling your body, right, which is our immune system, but they need an area where they can survey what is going on. I always kind of think about the lymphatic system as like the overpass of a highway, right? The lymph and all the cars on the underpass, right? Those are all things the lymph is kind of bringing through the lymphatic system. Okay, and then every once in a while you see a bad guy, right? And the patrol car sends another patrol car out to catch the bad guy, right? All of those patrol cars and all the cops are the immune system, and you're flagging the different bad things going through that lymphatic system. So again, the lymphoid tissue really provides that surveillance vantage point for our immune system. Lymphoid tissues, again, largely comprised mainly of that reticular connective tissue, which you guys all learned about in AMP1. And there's really two types of lymphoid tissue. There's our diffuse lymphoid tissue as well as our lymphoid follicles. So within our diffuse lymphoid tissues, essentially the diffuse lymphoid tissue is unrecognizable. You can't really see it, at least histologically, what I mean by unrecognizable. There's no real clear features to it, and that's because it's super diffuse. Pretty much every tissue, every organ in our body has diffuse lymphoid in it. Okay. Now, typically that diffuse lymphoid will be found in, the lamina propria of the mucous membranes. Okay. So it's widely dispersed and distributed, there we go, throughout our body. Okay. Lymphoid follicles, on the other hand, have a very, very distinct shape to them. See, this right here is a lymphoid follicle. Okay, and what you can automatically see, and the reason it's a follicle, see how it looks nothing like this other region out here? And there's some very kind of unique characteristics to a lymphoid follicle. It has a very dark pink, or not pink, dark purple outer ring with a lighter ring on the, or a lighter spot in the middle. And this lighter spot in the middle is known as a germinal center. Okay. And essentially what this is this is a solid mass, okay, of packed lymphoid cells and reticular fibers. So this here is the intestines, okay, part of our digestive system. And dispersed through that, we have these huge clusters of lymphoid follicles. Now, the unique feature of this germinal center, okay, this is very specific to our B cells. This is where the B cells of our immune system are going to proliferate. There are no T cells in our lymphoid follicles or within the germinal centers, okay? So this is very, very characteristic of an important cluster, important for that B cell proliferation, okay? Again, they'll form throughout large kind of lymphoid organs, but you can also find clusters of these throughout the digestive system as well as in the appendix. And these clusters that look like this, that are not part necessarily of our lymphoid organ, these are called Peyer's patches. Okay. Now, a Peyer's patch and a lymphoid follicle, they're the exact same thing. It's just that typically, if you have these clusters in a lymphoid organ, they're called lymphoid follicles. If you have these clusters in a non-lymphoid organ, i.e. the digestive system or the appendix, they're called Peyer's patches. Not my rules, just what they do. Okay. Then we have our lymph nodes. This is the primary lymphoid organ of the body. Okay. All of these little teeny nodules within this picture, those are all those are all lymph nodes. And so they're typically embedded in a connective tissue. They're typically in clusters along the lymphatic vessels. You can actually palpitate some of the lymph nodes that are near the surface of the body. So for example, in the inguinal region, which is like down, like where the crease of where your leg hits your torso, you have that crease. Down there, you can usually feel a lymph node. Underneath your arm, in the axillary area, you can feel that lymph node. In the cervical regions, right, you go to the doctor and they kind of palpitate underneath your neck. And even sometimes when you're sick, you can actually feel those lymph nodes right here become enlarged, okay? So again, you can palpitate some of them because they're more superficial. So the function of lymph nodes is kind of twofold. One is to filter lymph. So essentially, as our lymph comes through those lymph nodes, the macrophages that exist within those lymph nodes will essentially engulf cellular debris. They will engulf and destroy pathogens. They'll activate the immune system. Okay, which we can see here, which is the second part of our lymphatic system. So once you kind of start to filter out that lymph, those pathogens that are basically caught in the filter will initiate the activation of those lymphocytes, which will then mount an attack against those antigens. Now, I keep on using the word pathogen and antigens. Okay, so when we talk about, I don't know what I just did here. Okay, yeah, so I think I hit to tell me more, and I don't even know what happened then. Because we don't really need to know more. We don't want to know more. Okay, so when we talk about a pathogen, this can be a bacteria, a virus, a parasite, okay? Typically, a pathogen is anything that causes, oh my gosh, that's horrible, it causes disease. Okay, I'm on this little teeny desk, so my writing is really horrible. Okay, now, if we kind of designate this, like this little thing as a pathogen, we'll draw a little X like this, okay? On the surface of these pathogens are their antigens, just like on the surface of our red blood cells, okay? So I will often interchange kind of this notion of a pathogen and an antigen, and that is because if we're going to elicit an attack against the antigen, which is on the pathogen, we're eliciting an attack to the pathogen as well. Okay? Okay. So when we look at the structure of our lymph nodes, okay, they vary in shape and size, but most typically have kind of a kidney bean shape to them. The internal structure is actually divided up into what we call trabecula. And these trabecula kind of are connective tissue compartment, it basically compartmentalizes the different clusters or cortex regions within our lymph nodes. And so what we have is our cortex, which is this outer region out here, and then we have the medulla, which is this inner region there. When we look at the cortex, that cortex contains a ton of germinal centers. And again, when you hear that word germinal centers, you should automatically think of B cell proliferation. Okay, again, dividing. So within this lymph node, we have a massive amount of B cells dividing. And again, that's a good thing because we need those to elicit our immune response. We have dendritic cells that will also be hanging out there to help us activate that immune system. And down towards the bottom of this cortex, kind of down towards the base of this triangular, or the apex of this triangular system, you're going to have some T cells. Okay. Now, the T cells aren't necessarily housed just in our lymph nodes. The T cells are more likely to be circulating through our blood, our lymph nodes, as well as our lymph as well. Okay. Our medulla are basically just these cords that extend inward. They contain both B and T cells as well as plasma cells. Again, we'll talk more about plasma cells next week, but B cells will differentiate into plasma cells. Plasma cells will produce antibodies. Oh my gosh. Okay. As well as we have these what's known as medullary sinuses, and these will contain our macrophages. Now, there's a unique design to lymph nodes. And if you look at kind of how a lymph node is built, or how it is formed, what you're going to see is that you have this very large, convex portion to it. And what you're going to see is you have a lot of afferent tubes coming into this convex structure. Okay, so what happens is as all this lymph enters into this lymph node, okay, because if you look at where this lymph is leaving, right, you have this convex area here with only 2 vessels leaving. So what that means is all this fluid coming in is going to basically build up, or basically get backlogged in here, okay, in this lymph node, okay, and kind of form like a stagnant buildup. Right? So if you have like 5 waterways coming in and you only have 1 water or 2 waterways leaving, right? Sometimes you're going to get some flooding of that area that's bringing all of that fluid in, right? Now, in the lymph nodes, it's actually a good thing, because what we need time to allow for our immune system to activate against any pathogens that may be there. Okay. So this kind of design is actually a good thing. Because if it was a 1-for-1, right, that fluid would just fly through that structure, and the immune system wouldn't actually get any time to kind of see what was going on. So what we do is, by only having 1 or 2 efferent vessels, you slow down the rate that the fluid and the substance can move through this lymph node. So another structure we have within our lymphatic system is our spleen. Okay, the spleen is the largest organ, our lymphoid organ. It's served by the splenic artery in the vein, and it enters and exits at the hilum or the hilum of the spleen. And the function of the spleen is actually twofold. One, it's a site of lymphocyte proliferation and immune site surveillance and response, which is kind of like what lymph nodes are as well. But the other thing that the spleen will also do is it actually cleans our blood, okay? Which becomes, as those red blood cells start to die, we know red blood cells from chapter 17 only exist for a certain amount of time. They'll clean out the blood of platelets and macrophages, will move some debris. So it not only activates our immune system, but also cleans our blood. And I don't know why this keeps on coming up. Okay. So again, because it has two functions, the structure of the spleen will be characteristic of two different functions. So within the spleen, we actually have two distinct areas. One area is called the white pulp. The other is called the red pulp. The white pulp actually surrounds the central arteries. So over here in this picture here is our arteries coming up, and what we can see is this kind of lighter region here. This is called our white pulp. Okay. Now, this is mostly lymphocytes within our reticular fibers, and it is involved in immune function. So the best way to remember white pulp from red pulp is when we talk about immune function, we're typically talking about white blood cells. Okay. So white and white. Okay. Red pulp, which is all the rest of this kind of darker red area. Okay. This is where we're going to recycle those red blood cells, where we're going to get kind of recycle the platelets, and where basically we're going to phagocytize some pathogens. So this red pulp, I always remember it because it's really involved in red blood cell kind of disposal and recycling. Another structure we have within our lymphatic system is our thymus. Okay. Our thymus, again, is a structure, it overlays the heart. So in infancy and early childhood and early into their kind of teenage years, the thymus actually runs kind of from mid-neck region. It comes down and actually overlays on the heart, kind of like you see here. Okay, it actually overlays on top of the pericardium of the heart. Okay, however, as we start to age, okay, as we age, not we start, right? This will begin to atrophy, okay? And what atrophy means, if you guys don't know what atrophy means, is this essentially means it starts to shrink, okay? So by the time we're adults, it is a super, super small structure, okay? And what the purpose of the thymus is, okay? Was I saying thymus this whole time? Hopefully I was saying thymus this whole time. Anyways, what the function of the thymus is it's going to generate our immunity. So essentially what the thymus does is it produces our memory. Okay? Not our memory in our brain, but memory in terms of immune response. So when we talk about immunity, okay, this is all going to occur in that thymus. So a child gets a vaccine, right? That vaccine will generate immunity. That immunity is created within this thymus. Okay. This is why it is harder as we age to generate immunity, because the thymus is so much smaller than it was when we were children. Okay. So in addition to that, we also have our, or not in addition to that, in addition to kind of the structure and where it's located, okay, if we look at the thymus histologically, we have our cortex and our medulla as well to our thymus. Okay, the cortex on the outer region contains rapidly dividing lymphocytes and some macrophages. And the medulla contains fewer lymphocytes, okay, but it involves or contains cells that are really important called regulatory T cells that prevent something called autoimmunity. So autoimmunity is when the body basically starts to attack self. So T regulatory cells help prevent that autoimmunity from occurring. And the thymus, again, is different than any of the other lymphoid organs in the body, in that it has no follicles. So if it has no follicles, right, we know follicles are really important for our B cells. So our thymus doesn't contain B cells, okay? And the thymus itself does not directly fight antigens. So I just told you the thymus was critical, essential for generating immunity, okay? However, the reason it is essential for creating immunity is not because this is a place where we generate or fight our pathogens, but where we essentially teach our T cells how to fight. Okay? So if you don't have a thymus, you can't generate T cells. And if you can't generate T cells, you don't have an immune system. Okay. In addition to that, the thymus is different in that in pretty much all of our other lymphatic organs, the scaffolding is that reticular fiber, the reticular connective tissue. In the thymus, that's not the case. In the thymus, it's basically the stroma of epithelial cells that are going to basically provide that support to our T cells as they become mature in that thymus. So the last few things I want to talk about are something called MALT, which is mucosa-associated lymphoid tissues. You'll find lymphoid tissue in the mucous membranes throughout the body. Again, I kind of alluded to this towards the beginning of the lecture when we talked about that diffuse lymphoid tissue and lymphoid follicles. It protects the body from pathogens as they try to enter into the body. And again, you'll have large collections of MALT in the tonsils and your Peyer's patch as well as the appendix. Okay. You'll also find MALT in the mucosa of our respiratory and the genitourinary tract or organs, as well as the digestive system. We have our tonsils, which is really the simplest lymphoid organ. They form actually a ring of lymphatic tissue around your larynx. You have your palatine tonsils, which are located back here towards the posterior aspect of your oral cavity. So here are your palatine tonsils. You have your lingual tonsils, which are located here, kind of at the base, kind of back portion base of your tongue. You have your pharyngeal tonsils. These are the posterior wall of the nasopharynx, and then you have your tubular tonsils that are surrounding the open of the auditory tubes in the pharynx. If you've had your tonsils and adenoids removed, okay, what you have had removed are your palatine tonsils. And I don't know why, but they call the pharyngeal tonsils your adenoids. So if you've had your tonsils removed, those two top populations of tonsils tonsils and adenoids removed, those two are populations of tonsils have been removed. Okay. Now, the whole purpose of the tonsil is actually to gather and remove pathogens from our food in the air. And how it does this is, first, our tonsils contain these massive germinal centers. So all of these dark purple structures, you guys, are germinal centers, or not, are lymphoid follicles. And in the middle, we can see these light purple areas, right? Those are all germinal centers. That means we have this massive amount of B cells proliferating in our tonsils, which is crazy to think about. But essentially what happens is, as food and air kind of passes along the surface of this tonsil, these tonsillar crypts here, these structures here that basically form deep down crevices or crypts within the tonsillar structure, capture or catch kind of these pathogens. And what happens is as these get trapped in there, they get destroyed by our immune system, but they also elicit an immune system response. I think the last thing I want to talk about are our Peyer's patches, and these basically are just aggregates of lymphoid follicles. Again, I told you before, if you have clusters of lymphoid follicles that aren't in a lymphoid organ, they're typically referred to as Peyer's patches. Okay, typically you'll find Peyer's patches in our lymphoid follicles, or clusters of lymphoid follicles that we find in the distal portion of our small intestine. And again, we find very similar structures in our appendix as well. The whole function of these lymphoid follicles is to basically help elicit an immune response. It basically destroys the bacteria, preventing them from breaching the intestinal walls, and well as helping our body to generate that memory. That's really important for our Bs and our T cells.